Hi, it's October 31st, 2023, and we're here at CCNY at the Chemical Engineering Department, and we're going to talk with Professor David Rumschitzki. David, thank you for joining us today. And
thank you for coming and doing this.
So we'd like to find out about your path of getting here. What was your experiences in winding up as a professor here?
That's a good question. Well, I was an undergraduate. I started out as an undergraduate majoring in math and thinking of doing artwork as a second major. And then at some point when I was, they stopped, where I went to school, Cooper Union, they stopped granting degrees in mathematics and said if you wanted to graduate, you'll get an engineering degree. So I got an engineering degree and finished a math degree. And when I went to graduate school, I worked on mathematical problems in chemical engineering, including mainly proving theorems, so I was still pretty mathematical. And when it came time to graduate, I was sort of hooked on research. I sort of loved doing problems where you didn't even know what the question was, much less the answer. And using mathematics to formulate problems and to try to solve them, I found just lots of fun and really interesting. Then when it came time to graduate, it was, I could have gone to industry, but I wanted to continue working on research problems. I did a postdoc in Germany at the Max Planck Institute with Manfred Eigen, who was a Nobel Prize winner in chemistry, and I was working on mathematical theories of evolution, which was a way to get me more into biological problems. But then when I came here, I really gravitated towards problems having to do with fluid mechanics and interfacial mechanics. And part of that was I did I had a summer job at Mobil down in Paulsboro, New Jersey, and I was asked to look at problems having to do with trickle bed reactors. So that sort of set me on a path for quite a while looking at interfacial mechanics. And eventually I also got back interested in problems in physiology. In particular atherosclerosis and also more recently cancer. Is that detailed enough?
No, that's fine, thank you. And so
feel free to follow up on anything you want more clarification.
So did you so now here in your current research now is a lot of biologically related chemical engineering interface. Can you tell us something about that?
Sure. The great thing about chemical engineering is that we have a lot of tools in chemical engineering that were developed or acquired for problems having to do with productions of chemicals. But those tools are useful for many other things. And there are many problems, for example, in physiology, where when biologists look at the problem, they look at it from one perspective. They look from the perspective of individual molecules, proteins, what they do. Even looking at a whole cell is already a big problem for biologists, whereas chemical engineers usually come and look at a problem from starting out from the overall mass balance, the overall balances, starting from the large and working your way down. And those two approaches can really complement each other. And each one can lead to insights that the other doesn't have or doesn't normally see. So, I was at a meeting a few years ago, a workshop, where there was someone from Novartis who had some data on cancer that he didn't really understand and asked for some help. And so I was looking at the various types of models that existed, and there were models that really didn't have much biology in it, that were just sort of fitting curves of tumor size versus time and extrapolating, which are quite useful. There's nothing wrong with them not having biology in it. They have very few parameters, but they don't really give you much insight. And then there were other models that were much more focused on extreme details rather than looking at them overall. And there was one model, only one, that took an overview model. It was of a Japanese group, but they were so concerned in getting nice solutions that they only allowed tumors to grow and metastasize and not shrink. So there was no notion of treatment, there was no notion of immune response. And of course, it was a very nice paper, and it's been referred to many, many times. But what it was missing was the effect of treatment and how that interacts with tumor growth and metastasis, as well as the effect of natural immunity, in particular today, where some of the best new treatments for some cancers involves activating the immune system, called immunotherapy. So initially we thought we were doing a minor modification where we took the Iwata model, this Japanese model, and we added the notion of immunity. I should also say that rather than the previous models would look at a particular tumor cell or one tumor and try to predict what's going to happen to that tumor with lots and lots of details, that isn't always a good idea, because you could have 5 patients with similar tumors and each one does something different. So there's a certain amount of statistical variation. Instead of focusing on an individual tumor, we, and also the Iwata paper, took the approach of statistical mechanics of looking at an ensemble, a very large number of tumors, and looking at the size histogram, the number of tumors of each size, in not just one patient, it could be a population of patients, and trying to describe how that histogram changes with time. And that naturally, you try to get essentially the mean behavior of this tumor population, and you also get some information about the expected standard deviation. Now, when you only have tumor growth, it's no surprise that all the patients die, that all the tumors grow and metastasize. It turns out, what we found is when you put in immunity and or treatment, you get a really interesting interaction between growth and shrinkage. And that leads to, in addition to a natural movement of either killing off tumors or tumors growing, besides that, you also get effectively a one-dimensional random walk in tumor sizes that leads to a diffusive phenomenon. And it's the diffusion that spreads out the tumor size distribution, the tumor histogram. And without going into too many details, what this does is it gives a potential answer to a very important question in cancer. Now, if let's say you have a patient that is diagnosed early with cancer and there's a good treatment, well, there are 3 possible outcomes. One is the patient is lucky and the treatment works, the cancer goes away and never comes back. Another is the patient is very unlucky, and either the treatment doesn't work and the cancer doesn't disappear, or it disappears but comes back within the first few years. And those are no big surprise. The surprise is the third possibility, which is a patient appears to all medical scans to be cancer-free for 5, 10, 15, even 20 years, and then it comes back as the same cancer. And that's the part that is sort of mysterious. What was the cancer doing? Did it fall asleep? Why did it wake up? And at least that's the way the biological cancer literature sort of looks at it. What we found is that this interaction between shrinkage, say due to immunity under treatment, and growth can lead to a certain size regime where instead of the tumors growing or shrinking on average, they just bounce around, they just diffuse. And it gives, naturally, without this sort of formulation, suggests a mechanism for all 3 of those outcomes. That is, if you're above this regime, that the sizes are above this regime where tumors grow faster than they shrink, that's the first case, the case where the patient's unlucky. If the tumors left after treatment are below this regime where they shrink much faster than they grow, that's the lucky patient. But if you leave tumors in this regime that's diffusion dominant, where diffusion is not very fast, but it's still dominant there, then the tumors can bounce around in that regime below a level of detectability for many years, until they eventually bounce out, either on the low side and you don't see it, or on the high side in which it comes back. Now, of course, as Sidney Brenner, who was a Nobel laureate, who was the president of the Royal Society, had once said, ideas are cheap. You need data. So we teamed up with a group at Memorial Sloan Kettering across town, that has now moved to Oxford, since moved to Oxford. And they had this wonderful system. It turns out that zebrafish, little fish that people have in their fish tanks at home, are a great model for melanoma, because the stripes in the zebrafish are melanocytes, the same as in our skin. And they can get sporadic melanomas just like humans do, with similar metastasis, with similar mutation patterns. The other nice thing about it is between the stripes, the zebrafish are basically clear. So what my collaborator had done is he bred the fish to get rid of the stripes. So it has basically clear fish, slightly pink tinted, and he put a green fluorescent protein tag on zebrafish melanoma cells. So when you inject these fish, clear fish with bright green tumor, you can get data without killing the fish, because you can just take pictures every few days, and you could see as the tumor grows, as new metastases spread out, and you could also see when immunity kicks in and starts shrinking those tumors. So you could get many, many data points from each fish, and this was almost tailor-made for this theory, this experimental system. And we have got enormous amounts of data, separately for males, separately for females, separately for immune-compromised fish that we irradiated them, and separately for fish that were immune-competent. That way, from each data set, we only needed to get one parameter. We weren't getting multiple parameters from each data set. And we came out with some predictions that we were able to then do new experiments and verify them. So we think we really have a good handle on the melanoma in the zebrafish. And one thing we found, for example, was predicted that we were able to verify, was that the females, there'll be a range of tumor sizes where the females can eliminate the tumors and the males can't. Because as it turns out, from what we found, the female immune system was more robust than the males. And then we looked in the literature for humans after this, and we found, in fact, this is also true in humans, that what's called the mean mortality ratio, the fraction of females that survive X years after diagnosis versus the fraction of men that survive X years after diagnosis, for skin cancer is more than 2. It's more than 2 to 1. It's like 2.5 to 1. And for many cancers, the mean mortality ratio for females is higher than for males. So we didn't know this when we did those experiments, but we were very happy to see that what we found in fish does not seem to be totally irrelevant in humans.
What
and
about
now we're working
in
on
humans? A lot of the melanomas are apparently caused by excess
sun,
UV light. Is the
could also be for. Well, I'm not sure people have studied the source of melanoma from fish, but certainly if they're out in the open in the water, that would in a fish tank, that's not going to be the case. But if they're out in the wild, that certainly could be the case. The UV light will affect their melanocytes like it affects our melanocytes. And now we're working on a model of breast cancer in mice, which turned out to be an excellent model of triple-negative breast cancer in humans. The mice, unfortunately, are not see-through, so unlike the fish, so we're using very modern MRI techniques to detect metastases in the lungs that 8 or 9 years ago you couldn't detect because MRIs were chronically cloudy and imprecise in the lungs because of the presence of air, but new pulse sequences have made that possible. And I should also mention that there's a lot of mathematics in this as well as experiments. There's a lot of work we can do analytically to predict, for example, the probability as a function of the size of the tumor, the probability that it's going to recur, as a function of size and the expected time of recurrence. And that's important because if the expected time of recurrence is 50 years, well, then maybe one might opt not to overtreat, because the body has something's been discovered in the last 10 years, something called host response. And host response basically means that the body is not always that smart. When you go and you kill some tissue, tumor tissue, the body doesn't always know that you were doing it a favor. The body thinks it's been injured, and there's a lot of data showing that it can then reorient its structure to rebuild what was destroyed.
Yeah.
So if you overtreat, you could encourage host response. And maybe in some cases it's better not to overtreat. But you need to have a sense, is it likely to come back in 5 years? Or is it only likely, if it's going to come back, it's likely to come back in 50 years? That's really valuable information. Also, what the theory suggests, if it really pans out, is if you have tumors that are likely to come back, what type of treatment could you do to make them less likely to come back? And that's all the theory instructs you how to do that. Obviously, if we can show that this theory also applies to human cancers.
That's fascinating. Because personally, I have seen cases where two of my friends had cancer that was apparently quiescent for a number of years. They saw it on some scan. They went in there and did some cutting, and then all hell broke loose. That's exactly what you just said.
Right, and there at least those tumors were visible on scans. The scans are usually limited to roughly a millimeter or a fraction of a millimeter tumor. That's still millions of cells.
Yeah.
Plenty of people have tumors that are below that size, so they appear cancer-free, but it could be a ticking time bomb.
So, in the very interesting coalescence of engineering and biology that I think has probably been underappreciated in the past. So, what do students actually do in the lab?
Well, there are usually two types of students: the theoretical students and the experimental students.
Right.
Theoretical students will come up with analytic either analytic methods to solve special cases or approximate methods to solve the general case, and also numerical methods to actually do the solutions, as well as other formulations of the theory and extensions of the theory. The experimental students, and sometimes people do both. I encourage students, even if they want to do mostly one, they should do at least a little of the other, because it really it makes them a much more well-rounded engineering scientist later on. And of course, the experimentalists are the ones who have to do the experiments on either the fish or the mice, take the data and analyze the data. And the data analysis, turning pictures into numbers, is not trivial. And there's usually quite a lot of thinking that goes into it, so that, because in the end of the day, data need to be reproducible. So there needs to be a consistent way of quantifying things that is reproducible.
Okay. So your students can be experimentalists largely, they could be theorists, they could be some combination of the two?
Exactly.
Okay,
exactly. I encourage them to be some combination of the two.
And I don't know how many years you've been working at this interface, but have you had any students that have graduated that have been involved in these programs? Or is
Sure.
Okay.
I have, for the cancer work is not that old. I have one student who graduated about a year and a half ago, maybe a little more. And he's working at a company called ASML, which is a Dutch company based in the United States, in Connecticut, that is the company that produces the machines that make chips.
Oh, I know that.
That make computer chips. Yeah. And they're, I believe, the leading company in the world on that.
Yes.
And this student is really very good at both theory and experiment. And when they have problems or trying to figure out what's going wrong, he's on a small team that diagnoses these problems and tries to
the biology fell out, the chemical engineering.
The biology fell out right now. We're not sure how many years it'll stay there. I've had a number of students who graduated on a previous project which isn't necessarily dead. It's just one I have to revive a bit. It's sort of sleeping, is a problem on atherosclerosis. And I have various students who graduated on that. One of them is a professor down at BMCC. Another one works at P&G in, where is P&G? It's somewhere in the Midwest. One of them is a professor in Taiwan. So some of them tend to be professors. Not all, some do go into industry.
Yeah. Yeah, there's an interesting blend in the faculty. Some faculty members have a lot more
Industrial
industrially
experience.
students that go to industry. Some have more that go to its academic. It's interesting.
But just because they worked on a biological problem, as you can notice, the student who went to ASML is 100% a chemical engineer, and he's working on chemical engineering problems there. He may come back to biological problems or may not. Just because one works on a problem with a certain application doesn't mean that, from one's PhD, doesn't mean that one's going to be doing that the rest of their lives. Right. More importantly are the techniques that one uses and the flexibility that when one has a problem, one can figure out what techniques one needs to use and then learns them. And it's that flexibility that is really important. The analogy people always give is of hammers and nails. If someone only has a hammer, then every problem looks like a nail. Yeah. But if one is more flexible, then sometimes you need a wrench, sometimes you need a plier. And those are the really good scientists that don't just have one technique that tries to fit every problem, but is capable of looking at a problem and saying, I need to learn a new technique to do it, and they're not afraid to do it.
Very wise words. Thank you. Okay, so we're getting towards the end of the interview. I just want to ask you the following. If there are some students watching this that are thinking about possibly coming to CCNY, perhaps working with your group, can you give them some advice or?
Well, be prepared to work on new problems you never thought of. I think it's good to be open to things that are interesting. The other thing I would say is, and I usually say this to students, is that life's too short to work on to spend your life working on things that you don't enjoy. These days, particularly since university tuitions are so high, the main thing on people's minds when they go to college is, how will I make enough money to pay back my loans and to have a decent lifestyle? And whereas that's very important, there usually isn't a unique answer to that. There may be many answers. And amongst those many answers, it's worth investing the time to find something that you will love getting up Monday morning and going to work. Yeah. Because you're going to be doing that for much of, for the next 40 years. It's going to occupy 2/3 of your waking life. So why not find something that you're excited about?
That's great. Great advice.
I can't say I was that smart when I was a student. I think I just went with the flow.
And sometimes you're just lucky and you fall into something you just like. And
right.
Then, yeah. Well, we have this discussion many times. The students, some of my colleagues, for example, they start out in engineering, they want to go into business, they want to be managers. Then there are some of us that just started out in a particular area. In my case, it was material science, solid state chemistry, and they're happy doing that for their whole life. And there's interesting how different people react to this. Life changes differently.
Right.
Are some of us too rigid, or are they just too driven to make money?
Well, I think what happens is, at some point, fairly early on, a certain number of years after one graduates, one adjusts. As a student, one usually has a lifestyle that's not very expensive. Afterwards, within a few years, people start to expand their lifestyle to the point where it requires a certain income. And at that point, So at that point, it's very hard to change careers because usually when you change directions, there's going to be a period of time where your income drops. And if you're at the stage where you can't imagine a few years where your income drops, you stay in the same thing forever. And I know people like that. New York is filled with Wall Street types and lawyer and real estate types and lawyer types, many of whom are counting the years to retirement when they're in their 30s.
Yeah, I know those type too.
And usually people in science or in research and engineering, they don't even want to retire. They don't even think of retiring. They just keep going because it's fun, because that's what they like to do. And isn't that a nicer way to spend your adult life?
I couldn't agree more. I think that's good advice for some of the young ones. Okay, well, David, thank you so much for spending time with us today and giving us an insight to you, to your work, and to life here. So thank you very much. Okay.